we got your questions and we're going to answer some of them, but some of them we're going to follow up with you guys in an email, so don't worry about that. So I guess we're at the noon hour, so we'll go ahead and get started, but feel free if you're just joining us to go ahead and leave us a message in the chat to where you're joining us from, but we'll kick off the program. So if you haven't joined us before, hello, good morning, good evening, good afternoon from wherever you're joining us from. My name is Laura LeBur, and I'm a marine science educator at the Smithsonian Marine Ecosystem exhibit. Welcome back to the fall series of Career Dives Conversations in Marine Science. This series will continue to this is the last one, but we'll continue to highlight the career tracks, interests, and projects of marine science professionals working with the Smithsonian Marine Station and the Marine Ecosystems exhibit. So, Tiffany, thank you for joining us. Maven Academy from Missouri. We've got West Craven Middle. Hello. So many schools today. Thanks so much all for joining us. As everyone joins the webinar or has joined, I'd like to point out some of the features. So you can use the Q&A box at the bottom of the screen to submit your questions at any time throughout the program. We'll try to get to as many questions as possible, but please be patient with us as there may be a high influx of questions and we may not be able to get to all of them. There's another educator, Erin, on in the background, and if you see a message in the chat box, it will be coming from them. Throughout the program, you can use the chat box or the Q&A to ask us questions, but please keep the questions and comments appropriate and on topic. They are visible only to Smithsonian staff. This program is going to be recorded and will be live on Facebook and will exist on our YouTube channel, so you can access it anytime you like. But it's going to be, and it's also going to be about an hour. So I think that clears up a lot of the housekeeping items. I'll just say hello to a few more people and then I will introduce our fabulous panelists. So we have Rebecca from Maryland. Good morning. Oh, I think that's everybody. West Craven Middle, I said hello. All right, so we'll go ahead and introduce our panelists. So this session today features Dr. Blake Ushijima. Blake began his research career by studying coral disease in the Pacific Ocean, and he now studies the bacteria responsible for stony coral tissue loss disease, the outbreak in the Atlantic Ocean. Today we'll be talking about his research and the probiotic work his lab is doing to help treat and protect these endangered corals, and how he got to the Smithsonian and his journey so far. So Blake, it's great to have you join Career Dives. Welcome.
Thank you. I'm happy to be here.
So we'll go right ahead and get started. If you want to share your screen and take us back to the beginning of your career and how you got interested in marine science.
Sure thing. Let's go.
Perfect.
Well, to start from the beginning, I have to go back to where I'm from. So, So I'm actually was born and raised in Hawaii. So that little spot right there, you can imagine, surrounded by an entire ocean. There is some connection to the, the sea life. But just for a little context for people who may not realize is that, growing up in Hawaii, you're actually fairly far from the United States. So the distance from Hawaii to San Francisco is actually about the same distance it is from the Smithsonian Marine Station to San Francisco. So you can kind of see here, you're kind of isolated. But, growing up in Hawaii, not too bad. Some people call it paradise. It is a very nice place. Some of the best beaches in the United States are in Hawaii, and you're always maybe a 15 to 30-minute drive away from the ocean. Here's a couple of pictures I've snapped over the years. It's one of my good buddies jumping off a cliff on one of the beaches we used to go to a lot. There's abundance of marine life all around. So, sometimes you see a couple turtles, some sharks, some interesting corals, and really some of the best sunsets I've seen. And, ever since I was a little kid, there's a strong connection to the ocean. You're always going to the beach. My mother used to take me to the Waikiki Aquarium a lot, see their little exhibits. Just, hanging around the beach, me and my sister hunting for sand crabs. This was really cool. This was actually the blowhole on the island of Hawaii. It's a collapsed lava tube. They have them around the island, so when the waves come, they'll fire up water jetting out of the ground. So that's pretty
Get
cool.
rained on by that one? Does it sprinkle on you, or you could just?
Depends where the wind's blowing. I wouldn't get too close. The force is actually quite strong, apparently. Never want to test that. So, but I mean, when it's really strong current or really strong waves, you can see it firing up fairly, high in the air.
Wow.
Of course, this was like maybe 6-year-old me, so everything seems really, really far up. But my real inspiration was actually my grandfather. So he was a lover of the ocean. He was an avid diver. He used to take me to the beach almost every day. This is me and him. This is on, little old, maybe 4-year-old me crawling along the lava rock beach. And this is me about to go spearfishing with him. So he had about a 200-gallon saltwater aquarium that I helped him, put stuff in, and you can stare at and watch all the little critters. But actually, one of the big pastimes was just collecting shells and things from the beaches, because he was also an artist, and these are some of the murals he created from these shells and things collected. This one in particular, I remember I physically remember helping him, arrange. Well, not I don't know how much I was doing, but I was helping him sort shells. But some of these were quite large. I think this one's about 3 or 4 feet across.
Wow.
So that's really where inspired me to really love the ocean, and I always thought it's like, well, maybe later on I'll have a career that involves this. And lo and behold, I did. So it actually started while I was an undergraduate at University of Hawaii at Manoa. I started interning at the Hawaii Institute of Marine Biology. And this is a cool little marine station because the entire marine station is on its own little island called Moku, or Coconut Island. You can see right here. And fun fact, this island was the opening shot they used for Gilligan's Island. So this is worked on Gilligan's Island. But it's quite nice because it was in a little semi-enclosed bay. So the lab wasn't too bad. You can see this is our picture of our lab here.
Step right out the door and snorkel.
Well, this was, you got to walk across a bridge. Actually, right outside our front door was the shark pen. So you can see here, this is my neighbor. It's a little tiger shark. But then you walk out over, this is more toward the edge of the island. You can see this lighter area. That's actually the fringing reef. And you can see the just full of coral. The really cool thing is, like, here's a shot that almost the entire bay was bordered by the Ko'olau mountain range. So, the view wasn't bad. Sometimes the water would be pretty calm. You can kind of see the fringing reefs around the island, some of the coral reef around the station. So, it wasn't usually too difficult to find coral and have some really nice photo ops as you're working.
Those corals
on your lunch. Yeah, so this is one of the dominant corals, the Montipora capitata, the rice coral. It comes in these two very nice colors, the brown or orangish color. But while I was working as an intern, taking care of tanks of corals, or doing a bit of sequence analysis for the coral microbiology project. This is really learned a lot of the basic things that I carried on throughout my career. So this is Ashley Smith. She was the graduate student that was my advisor during my internship. Pretty much where I learned most of the things like I use today. And it was also under Dr. Greta Abbey. She's a coral disease ecologists, probably in my opinion, one of the best coral disease ecologists, but I might be a little biased. Yeah, I mean, it was a great experience. Got to actually see the disease up close. Back when I used to go into the field, you pop in and collect your corals, set up your experiments. I have to say it wasn't easy. You have to go in every day, make sure everything's okay. As a lot of marine biologists know, corals can be very finicky. I'm just to show like how important it is to take care of the environment and the effects of global climate change is like little small changes in temperature, light, salinity can be disastrous for experiments, but also applies to the environments they're coming from. So this is really, I really started to realize like, oh, this is something I really want to do. Like it's hard work, but this is really fun in my opinion.
Yeah. You get to do a little bit of everything: field work, setting up your coral in the experiments in the lab. There's really real world applications to the stuff that you're learning about. So I can see that it would be super fulfilling.
And so this led me to stick around for graduate school. So still co-advised by Dr. Greta Abbey, I joined the lab of Dr. Sean Callahan, and started off with my master's and then moved on to a PhD. I switched, master's was fun, but I was like, well, I feel like I could do a PhD. Seems fun. Really like the work. I mean, it's not bad working here in Hawaii. Had a really big lab, a lot of supported lab mates. It's actually some of them. I like people like me showing this picture because it shows me in our formal attire in Hawaii.
Flip-flops are very formal.
Yeah, this is for my one of my last interns when I was a grad student, Andrea Nzueta. She is now doing her PhD at Northeastern, and one of my lab coworkers, Dr. Sylvia Grant. This is a great picture of one of my other coworkers, Dr. Patrick Vidot. We used to go to a lot of conferences together. So it'd be us kind of schlepping around the country, carrying our posters. But yeah, a lot of great memories. So a total of 6 years over at 2 degrees. So it was really cool. Yeah, I mean, we ended up studying some pretty cool stuff. Interestingly, the main project of our lab was working on cyanobacteria. So these filamentous bacteria that can photosynthesize or use sunlight for energy. And our actually main expertise in the lab was genetics. So we looked a lot at bacterial genetics, how these cyanobacteria can differentiate in the different cell types. But really for layman's terms, we could do really cool things like make some of the cells fluoresce green. One of our coworkers, Dr. Orion Rivers, he managed to get a cover of a scientific journal. Think of a really nerdy way of getting on like People magazine. He got the cover of this scientific journal with his mosaic filaments. So you can see here these cyanobacteria are fluorescing 3 different colors. So really cool picture.
And it's a big deal. I mean, it's scientific to be on the cover of a scientific journal, like that's a huge, like you said, getting the cover of like, I don't know if People is a good enough example, but like Time magazine, that's a big honor.
Yeah, so we were really excited. I mean, it's also a cool photo. So. But the other portion of our lab, we studied coral disease. So this is some pictures of the disease I studied. There was actually a few in Hawaii. One of them is acute Montipora white syndrome. You can see here, this is the normal Montipora or the rice coral. Very nice color. But then they would get infected and you see this white right here. So that's actually not coral bleaching. is actually the exposed skeleton. So if they get infected, the disease would actually kill the living coral, which is really sad to see this. Here's actually a time-lapse video of how fast this infection can spread. You can see how this coral fragment basically dissolves away. So we're trying to understand, like, well, what's going on with this disease, what's causing it. Here's a nice picture of all the coral we use, so we could, collect little fragments and break them up in even smaller fragments. Acquired a little learning curve to actually do that, but over time, you eventually can perfectly break these corals into similar sized pieces.
And for those of you who don't know, fragmenting coral is a pretty common practice in coral research because they naturally will fragment and the pieces will grow into a whole new coral colony. So it's not just about breaking them into smaller and smaller pieces. These pieces will eventually grow, and the coral can regenerate that loss. So just to clarify, because I don't know, it's super common in coral research, like I said, but maybe there are people joining us who haven't ever seen that before.
Yeah, so some of the things we did work with coral, and it's grad school, not always funds available, so you make do with what you can. At the time, I was really really proud of the very small budget I had for one project and what I could do with some Walmart Tupperware. It was a lot of hard work. It wasn't all fun and games, playing in the water, sitting on the beach. Here's a kind of example of the seawater I used to have to haul for my experiments. That was one cart. You never realize how heavy seawater is till you have to move about 700 pounds of it. So that was one of the carts. But we bring it in, filter it, sometimes sterilize it. So I thought it was a really pretty picture, but this is one of the experiments I ran. The bacterial growth media changes color depending on the type of bacteria growing on it. So it was fairly common, 3 a.m, someone be in the building working on an experiment. And this is, threw this in because this is a very funny story behind this. This is one of the pressure cookers I used to bring into the field, or if the autoclave, the sterilizing unit at the university went down. And then this eventually turned into a paper that was published with one of my coworkers about using the Instant Pot pressure cooker as a cheap alternative to an autoclave. So that was published, and it was really great. And just one of these simple things that that ends up being a scientific publication.
But it makes it really accessible because anyone can get an Instant Pot and you can use it for
yeah. Yeah, an Instant Pot, it costs about $200 versus an autoclave, which will maybe run you about $1,000 for a really small one.
High on the low end.
Yeah.
That's so neat. It looks really, it looks like you guys had a lot of work to do, but you kept it fun too. I see Nicolas Cage presiding over some of your samples there.
Yeah, that was a weird instance. Someone had stuck a life-size image of Nicolas Cage's face in our incubator. So I took it upon myself to, it's like, well, I think it'd be funnier with googly eyes. So I stuck googly eyes on it.
He's just watching over everything, making sure everybody's behaving.
Yeah. Our advisor thought it was weird, but
hey, if they're 3 a.m. in the morning, at least you have somebody to talk to.
Yeah, I guess so. If you put it that way. But yeah, there was a lot of cool research going on. Just to actually, we had a lot of fun. A lot of cool research, but it was really studying somewhat of a kind of a depressing topic. So the big thing was in Hawaii, we had a few coral disease outbreaks that occurred in the same bay as the marine station we worked at. And you can see here is like one of the good things about Hawaii is they're blessed with, so there's some areas with very high amounts of coral cover. The bad thing about this is you have so much coral that if there's a disease outbreak, it actually helps spread the disease. So you could, it might be a little hard to tell, but the Montipora, the rice coral is normally this brown or orangey color. The white green is all dead coral. And you can see here how it kind of travels along from colony to colony, killing these corals. This is up close of one of the colonies. You can see here about half of it's all dead. It's a nice little video clip taken by one of my coworkers. She was swimming along, and it's, admittedly very depressing to see like how much dead coral there is. You can see how close they are, and unfortunately to help the disease spread. However, that's, we, all of us are like really working hard on this project. Made quite a few big discoveries actually in Hawaii. During my master's, I isolated this bacterium called Vibrio wentzii, and I confirmed it was one of these coral pathogens that can cause disease in the rice coral. I can see here it's a normal, nice bright orange color, but this is after infection with Vibrio wentzii. You can kind of see that has that white tinge. Well, that's actually the skeleton poking through, and it was a slow moving disease, but over time you can see it just kind of eating away at the coral tissue. But then we eventually discovered 2 more bacterial pathogens, Pseudomonas pyrotica and Vibrio cordylicus, that cause much faster diseases. So some of these will take only a few days. You have a small infection occur. This is the time-lapse video. That infection was caused by Vibrio cordylicus. But one of the really cool things is that one of these coral pathogens, Pseudomonas pyrotica, turned out to be a novel species of bacteria. And it's actually one of the only a handful of bacteria that can produce these very unique structures called prostocae. So they look like little tiny fingers that grow out of the bacterium. So it's a really cool little bacterium. And so you notice that it's called Pseudomonas pyrotica. So one of my coworkers who led the main work on actually defining it as a new species, Dr. Sylvia Grant, she was really into pirates. I mean, who's not into pirates?
Right.
So we named after pirates because, if you discover a new bacterium, you get to name it. So just, a little note to all the all the future scientists out there. Just, if you want to name a bacterium, just go out and try to find a new one, and you can almost name it pretty much anything you want.
Anything you want, except after yourself, right? You can't name something after yourself.
I mean, it's frowned upon. It might be a little pretentious.
I'm the best. I'm naming this after me.
Yeah. But, it's really cool. I mean, because we have a bacterium there, Pseudomonas pyrotica, named after pirates. So that's actually pretty cool.
So Blake, that's actually a really good segue because Dr. Melidio's class is watching, and they asked us, one of the questions that they sent along was, how many underwater sea creatures have you and your fellow marine biologists discovered? So you worked alongside some people who got to discover this new bacteria, and maybe it's not exactly what the class had in mind when they said underwater sea creatures, but bacteria also live in the sea. So
I mean, bacteria are underwater sea creatures. They're underwater and they're in the sea, and they're technically creatures. It's funny. This is I've been involved with the discovery of 3 new species of bacteria. One of them was during the marine micro class. The lab I helped teach at University of Hawaii. The project was just isolating bacteria and just seeing what species they are. One of them turned out to be a new species of bacterium. Then there's Pseudomonas pyrotica, and then I isolated another bacterium. I don't want to release a name yet because it wasn't published, but it was, we ended up naming it after one of my colleagues' daughters.
Secret bacteria.
Mostly because it was a cute little bacterium. But that was actually isolated from Palmyra Atoll, which is actually the next, topic of this presentation. So during grad school, a lot of the summers, I was lucky enough to go out to this field station in Palmyra Atoll. Just so you guys know, that's where Palmyra Atoll is. Not usually found on most maps. If you thought Hawaii was remote, this is even more remote. And this is actually a picture of the entire atoll. That little line there is the runway. So you come in on a tiny jet and you land on this runway, which is, the Palmyra airport is pretty much, a dirt strip. Here's the waiting room for the international airport.
Just the one chair.
One chair. Sometimes, someone needs a chair and sometimes it's not a chair. So you got to stand, sit in the grass. Beautiful place. It's a nice picture of the sunset from the galley. Most of the buildings are surrounded by this jungle, but they had boats. You can go out and do field work. But it's understatement calling it a beautiful place. This is how clear the water is in some areas.
Wow.
If you've ever been to a beach where there's absolutely nobody on it, that was really cool. One of the funny things is, I like looking at shells. I mean, couldn't take any of the shells, but one of the first things I noticed is if you walk along the beach, it's just littered with all these little shells. Well, if you look closely, all these little shells are tiny hermit crabs. So you really can't take them anyway. But it was really funny because you're walking along the beach and you notice, like, why are the shells moving? It's like, oh, just thousands and thousands of tiny hermit crabs.
Wow.
But speaking of hermit crabs, those are the baby hermit crabs. They actually can grow big, about the size of tennis balls, and they like to steal your stuff. So this is a nice picture of one of those hermit crabs. It's still a PVC end cap from our supplies bucket, and decided to use that as a shell. And in addition to these tennis ball size hermit crabs, there are what are called coconut crabs on the island. They're called coconut crabs because some of them can use coconuts as shells. This is my size 11 next to a medium sized coconut crab. So some of them got a little bigger than that.
Whoa.
So, watch where you step at night. But it was actually quite a few cool critters. This is a red-footed booby called Rain. A nice little seabird buddy that I made. Used to keep me company by the water tables. And this picture is, this is probably one of the coolest dogs I've ever met. His name was Dadu. He was one of 3 dogs that were left on the island that was previously owned by this rich couple before the Nature Conservancy took over.
Wow.
And what him and his 2 brothers did to survive is one of the things they used to do is hunt sharks. So right off the shore, you can see a lot of these small sharks. So Dadu and his brothers, this is an old picture of one of his brothers with the shark he caught, would literally hunt sharks to survive. That was their food.
That's hardcore.
Yeah, so really cool. He was very old when I first met him, but really cool dog, really chill. Just spend most of the days just lounging by the beach, looking out at the ocean, or maybe looking at sharks.
Scoping out the next meal.
But yeah, a lot of really cool things on land, but also a lot of really cool things in the water as well. So,
yeah, let's talk about the sea critters, Blake, because there was a couple other questions that were again, from Dr. Melidio's class. They sent their questions ahead of time, but if you're listening in and you want to ask some questions, please feel free to use the Q&A feature at any time and ask your questions, and we'll address them either as they come up or at the end of the program, wherever is applicable. But they had some questions that were like, have you ever seen a rare animal? What's one of the really cool animals you got to see underwater? And so I think now is a good time to talk about some of those cool critters.
Oh, there's a lot of cool things. I mean, besides the multicolored corals they have around Palmyra Atoll, one of them here's a giant clam. Well, it's a small giant clam, but they get quite large. This one was about the size of my head. A lot of colorful fishes. This is, just, I don't know what type of parrotfish. I didn't get a good picture, but also ran into what's called a humphead parrotfish, which grow to the size of you. So I came face to face with a parrotfish that was the same size as me. So that was quite exciting. Wasn't expecting it, but it's quite exciting. Saw a few sharks. Luckily, most of the time if you see sharks, they don't really bother you unless you bother them. But in this video, it might be a little hard to tell, but this is actually a giant manta ray. And this is right off our dock. So it's about waist high water. And they had a little tiny light by the docks, and the light would attract plankton. So most nights you have these giant manta rays come and just kind of circle around and feed. So at night you can just kind of sit by the docks, watching giant manta rays, which a lot of people can do. So this guy is probably about, I'd say at least 6 feet across. Huge, very gentle creatures. You don't really want to bother them, but really cool to see.
Wow, they're beautiful. I love watching them swim through the water. They're so graceful. Hopefully you guys can all see this video. There's 2 in that picture, Blake, or 3 in the video?
This is just one.
Oh, just one.
He was the most common one. Sometimes you'd get 3 or 4 that are coming around, just going in a circle. Really, really cool creatures. They're just kind of chilling, filtering out. You can see the little flaps, filtering in all the plankton.
That's awesome. Oh, so beautiful. Well, Blake, if you're up for it, we've got a few questions that have come in. We can take kind of a little break here and talk about some of them because they applied to some of the work that you did in Palmyra and in your graduate degree.
Sure.
Okay. So Natasha asks us, did you just study coral or did you study other things too? And maybe I should have asked that one because we'll get to that in a second.
Yeah, it was mostly coral during my graduate degree. When I moved on, I actually have a couple of pictures of it later on. I did study a couple other marine critters when I used to do work on aquaculture. But I have a bunch of pictures for those. But I worked on a bunch of different projects as well. So I didn't just do coral disease. I worked a little bit with, things like amoebas. I did some work with cyanobacteria. Did a project on, sewage microbiomes. A lot of random things.
So Natasha, stay tuned because we're going to talk a little bit more about that when Blake shares about his postdoc experience. Okay, so someone else would like to know, why did you choose to study coral?
Well, because it's such a big part of the culture I was growing up in Hawaii. I mean, the coral reefs, essentially where it kind of relates to like the forests of land, the Amazon rainforest. That's where all the creatures are. So if you think about the ocean, it's like the coral reefs, that's where you have a lot of habitat for a lot of sea creatures, a lot of reef fish, a lot of juvenile fish. And it's, the ocean is so important for Hawaii that, if you lose the coral reefs, you have this breakdown in the entire environment. And, one of the things is that I grew up, my grandfather liked to fish a lot. So, I mean, I always thought the oceans, like that's where, you can get dinner from. Um, and, everybody likes going to the beach. Everybody likes, seeing, beautiful marine life. So it was important to me. Like that's, it's kind of part of your home, you have to protect the environment. And that's what makes, one of the big things that makes Hawaii is the environment. So, that's kind of why I went into coral disease. And it's really cool. It's, it's really cool. So that's the other reason.
I mean, hey, it is really cool. Madison would like to know, I'm assuming about this is a question about the Palmyra Atoll, but what is the most, you talked about the hump-headed wrasse, but what is the most common fish on the island?
Well, the most common fish?
There's a lot of fish diversity there, correct?
Yeah, there's a lot of fish. I don't study fish, so I didn't really know. I mean, there is, that's a really good question. There was a lot.
Busy paying attention to the corals, all the fish are just background noise.
Well, I mean, it was like walking into a candy store. It's like all these different things around. It's like, oh no, that's cool. That's cool. That's cool. Actually, on this next slide, I have a nice picture. This is actually a school of convict tangs I ended up following for quite a while. I mean, there's a lot of those.
Wow. How many do you guys reckon are in that picture?
Oh, I was never
1,000?
I was never good at guess the amount of jelly beans in a jar game. So it's quite a few. I ended up actually accidentally swimming through it too, because they kind of took a sharp turn and headed right toward me. So I just stayed still, didn't really know what to do when you have a school of fish just swimming right toward you. Didn't want to freak them out. But yeah, I mean, there's a lot of fish. I mean, there's also a lot of coral. I mean, you can kind of see here some of the pictures I've taken, some of the really cool colors, bright orange, purple, weird morphologies. Some very large colonies. So this is for those who work on corals, the Probiotics colony. I think it was about maybe 8 feet across.
Wow.
We were looking at this area where these very large colonies. It's actually a underwater photographer from National Geographic. He happened to be on the island too, and then we're gonna go out check out the large colony. We're like, okay, cool, I'll go too. Need a hand? And you can see like how much coral is just all around Palmyra. There's a couple members of our team. So there was a lot of fish, a lot of things. Didn't really do much counting.
So to transition back, I guess, to talking about coral and to talking about your experience, I think this is a good slide to talk about it because you can see all of you working underwater with tanks on or free diving and snorkeling. But someone from Mrs. Fisher's class would like to know, did you ever get hurt studying coral?
Not seriously. You can just be aware of what you're doing underwater, be aware of the current. I mean, we've all been hit by a wave and thrown across rocks. So sometimes you get kind of beat up, scraped. Also, you'll learn to treat and clean out your cuts very well. I mean, corals are pretty, but they're also one of the things is they're covered with this mucus layer that has a lot of bacteria in it. So you have to treat coral cuts very and to take them very seriously. Make sure you clean them out very well. But and it's also about being aware of your environment. Don't just go and stick your hand into random holes. You never know if there's an eel in there or some other thing with sharp teeth. But yeah, I mean, there was a few injuries, but no, nothing too serious.
We'll take one more question here. Nana wants to know, do you remember the name of the National Geographic? Sorry, whoa, words, putting them in sentences. Do you remember the name of the National Geographic photographer who came to Palmyra? And then
Oh, wow.
maybe not.
That was a while ago. I don't really remember his name. It's kind of distracted too.
Yeah, I can imagine with all the stuff going on around you. But we have a lot of really, really great questions. Thank you guys so much for asking them. As they come through, we'll try and get to them, but we're going to move on and we'll take some a little bit farther down the line because you guys have a lot of questions about corals. But we have a couple more things to get to before we wrap up today.
First, the next is just kind of showing, it wasn't, a vacation. One of the things we had to do was we had to bring, Palmyra Atoll is not, there's no, Home Depot or Walmart there. So you have to bring in all your supplies. Like if you wanted to do work on corals and you had to build, our water tables, you had to build, your water tables. So Benji just shipped everything, all our supplies that we need. It was great help from one of the technicians, Roland Kano. Helped them, assemble it with it. I think we managed to assemble it in like 3 or 4 days.
Wow.
Which is, it seems like, oh, you just set up a couple of tables. Yeah. We also have to realize, Palmyra Atoll, the heat and humidity, it makes Florida seem okay. I think during the day, the surfaces would get up to like 100, 18, 120 degrees. So we get pretty hot and humid. But we got it done. A lot of hard work. And the big thing we're studying was that unfortunately there was a coral disease outbreak on Palmyra Atoll. It was killing off the table acroporids. So these large table corals. So this coral is probably at least 6 feet across. And you can see here, this is the living tissue that's remaining in all the dead skeleton. You can see this white is exposed skeleton. But the disease, you can see here, this is when the infection started, and 1 day later, you can see there's not much left.
Wow.
Would just eat away at the coral tissue, leaving the skeleton. And from my work and a lot of help from a lot of people, I was able to figure out it's caused by a bacterium called Vibrio coriolyticus.
So this is a good, sorry to
No.
This is a good segue because a couple people have asked like what is causing the coral diseases. So the bacteria that you're working with are what are actually like attacking the coral tissue and breaking and eroding that down, right?
Yep. For some coral diseases, there's a lot of different microorganisms or things that can cause disease, but some of these coral diseases are caused by bacteria. The same thing you see with other animals, and you can have bacteria or viruses or genetic, the environment causing disease as well. But I kind of wanted to show this because it's kind of the background that I don't really typically show is, we worked a lot on corals, looking at disease, but our expertise in the lab was genetics, and we did a lot of background genetics, trying to figure out how these pathogens are working. And I thought this was a really cool visual. So the disease in Palmyra, we found the water temperature affected if the bacterium was able to infect corals or not.
Wow.
So at higher temperatures, the bacterium would infect corals very well, but if the temperature dropped, it wouldn't affect temperature, wouldn't allow the bacterium to infect corals and cause disease. So one of my interns, Donna Pascablo, so I walked her through this. She was able to create, we didn't have money for a lot of this fancy genetics. So we took a somewhat cheaper but more complicated approach. So I walked her through creating this genetic system so that she can look at different genes in this particular pathogen, and if the gene was turned on, it would turn the bacteria blue. So we were working with looking at what genes were turned on at what temperatures to see what genes are responsible for the infectious process. You can see a couple of pictures here. Some of the genes we found at low temperatures. Some of them are white. At high temperatures, now the bacterium turns blue. So it's a really cool thing. So it's my intern Donna, and later Jack Stengel, who did a lot of the work on looking at the different isolates. So it was really cool. Some of the cool things with genetics, you can turn bacteria blue.
Whoa. The plates that you guys make are so beautiful. Like, I mean, it's kind of weird, maybe a weird thing to say, but all the different colors and the different ways that the bacteria respond and that they respond to treatments and stuff, they are quite beautiful.
Oh yeah, I mean, auger art is one of the hip things to do.
Is it?
Yes, so many pictures on the internet nowadays. You can see some really beautiful auger art, and some of these marine bacteria produce like a very wide array of different colors.
Wow. I mean, it is, that blue is like a beautiful blue.
Yeah, well, this is a genetically engineered bacterium, but some of the natural colors are still just as, bright purples, reds, oranges, pinks. I've had iridescent bacteria, it looks like kind of like glitter, neon greens, so a lot of different colors.
So cool. Oh my gosh, I like it so much. I just want to really quickly, since we're here, take a minute to address. So if you have any questions, we're not using the raising hand feature, but you're welcome to ask them in the Q&A. We have a lot of questions coming in today, which I really appreciate, but we're not going to have a time to get to all of them. So please make sure that your questions are on topic and appropriate for what we're talking about today. If you have any follow-ups, you can always email us at smseducation@si.edu, and we'd be happy to answer those questions there. I know we're getting a lot of them, so I just wanted to if we're not getting to them right away, don't worry, we will get to them if we have time. But Erin is going to be putting our email address in the chat for you. So if you're our teacher and your students have a lot of really amazing questions, please, if we don't get to them, please follow up with us and we'll do our best. So with that, we'll move back to bacterial genetics and what you did, Blake.
Yeah, well, speaking of cool bacteria, there was several other diseases that I worked on as well. A couple of collaborators and I worked on this fungal disease that affected what's called crustose coralline algae. So this pink algae that can form calcium carbonate skeletons like corals. And it was being infected by this fungus. Ended up having to take samples and slowly degrade them with a weak acid to actually expose the fungal filaments. So it's a couple of pictures here. We also worked on this other coral disease called black band disease on the island of Kauai. You can see here, it's very distinctive. It's this black band. And some of the culprits was actually a pathogenic type of cyanobacteria that was actually motile. You can see it that actually fluoresced red.
Whoa. And
another type is another motile filamentous bacteria called Vegeatoa that was part of the sulfur cycle. You can kind of see here slowly moving this bacteria, look like little tiny snakes as well. So you have all these tiny little wiggly snake-like bacteria in this black band you have here. And few of the other things I worked on. It was over the course of my master's and PhD, ended up working on a lot of quote unquote random things as well. But then when I graduated from the University of Hawaii with my PhD, I went to Oregon State University and started some work on aquaculture projects. So I had to work with things like Pacific oysters. You can see here, quick little setup with beakers with oysters in them. I also did a little work with Pacific shrimp. So this is when we got them, I think it was about a week or two old. And over time, they get a little bigger when you start growing them. Here are the few ones that were left over at the end that were our laboratory pets. So these are about 6 inches in size. They look more like prawns. But yeah, but a lot of my work, Oregon State University, this is where I started working a lot on probiotics, so beneficial bacteria that can help either oysters or shrimps or things like that. And a lot of it had to do with oyster larvae, because a lot of these oyster larvae can get these really bad infections and they can die from them. It's not as pretty as coral, but I still think it's really cool. But most of my experiments kind of look like this. Oyster larvae are really small, so most of them, they kind of look like little specks of dust in the water. But if you look under a microscope, you can see most people don't realize oyster larvae are motile, so they're kind of swimming around here. And like I mentioned, some of the really pretty marine bacteria, some of the probiotics we worked on, very nice colors. You can see here they inhibit the growth of other bacteria, these clear zones, that preventing other pathogenic bacteria from growing. So this is really where I started really focusing on the use of probiotics to help marine organisms. And this kind of leads into how I ended up at the Smithsonian Marine Station. One, because I used to work on coral disease. So my former advisor, Dr. Greta Abbey, started working with Dr. Valerie Paul at the Smithsonian Marine Station. I believe you guys had a career dives of her last year. Was it?
Beginning of summer, but yeah.
Beginning of summer. Feels like
feels like a whole lifetime ago.
Feels like a lifetime ago. Yeah, that's kind of where. So Dr. Abbey called me up and was like, "Hey, I want a microbiologist here. Need to help with this project." I'm like, "Sure, I'd be down." So it's kind of one of our initial setups, just me posting up in the visiting scientist lab with my mounds of plates, looking at what might be causing this coral disease, and this is where I kind of started working on potential probiotics that may be used to help fight this disease. And then a couple, came back for a few more summers. There's a couple of pictures of the team. A lot of people were part of this and helping, get this project going. Really didn't mind that Florida is, quite nice. Reminded me of Hawaii, so didn't mind too much. But yeah, it was, working with this team that eventually led to the Coral Health and Marine Probiotics Lab over the past year. We were able to set up this very cool system to house healthy corals, areas to work with diseased corals, and have a microbiology lab set up at the Smithsonian Marine Station where we could do a lot of work. And about this time, this is when I moved from Oregon State University to work full-time at the Smithsonian Marine Station, and ended up getting funding, hiring a bunch of really awesome interns from last year. I think the last career dives you interviewed one of them, yes, Marie Del Flor. Mm-hmm. Still at the Marine Station, kicking butt, doing a really good job.
Holding it down.
And this is kind of where we ended up. One of our probiotics seems to be working pretty good against stony crow tissue loss disease, which is currently doing a lot of damage to the Caribbean. It's kind of a mosaic of without the probiotics, you can see this coral eventually dies. With the probiotics, it seems to stop the disease in the laboratory. Currently testing in the field. But if you want to stay up to date, you can follow the Champ Lab on Twitter at Polyp Protectors. So I was there full time for about a year and a half. But then this past summer, I was offered a position at University of North Carolina, Wilmington, And so that's where I am now. I started as an assistant professor. And so the Ushijima Lab is a work in progress. It's coming soon, spring 2021. We're having a few students starting and having a bunch of equipment delivered. But yeah, we're actually going to continue the work on looking at developing probiotics to help fight core disease, and also trying to figure out what's the cause of the disease that's spreading throughout the Caribbean.
And you're going to still be working in partnership with the Smithsonian in the summertime, right?
Oh yes, yeah. I plan to can't get rid of me that easily. So I'm coming back. Yeah, it says thanks to Dr. Valerie Paul. She was keeping things running, and really great to work with. So yeah, I'm really excited to keep continuing the work on the project. So.
And it's so important too, because the stony, I'm not sure how many people are from Florida, but the stony coral tissue loss disease is really affecting a lot of different species of coral, and it seems to be very prolific. It keeps killing corals that have been around for many, many years. But
Oh, good.
But speaking of disease, we have had quite a few questions come through about can the coral bacteria also infect humans, or like what does that look like?
I guess I was talking about the coral pathogens, the ones that can cause coral disease.
Yeah.
The ones I work with probably don't infect humans. They can't live under the same conditions as a human body. So the human body is a little warmer than the ocean, and fortunately, some of these bacteria can't survive at that high temperature. But however, we treat in the microbiology lab, we treat everything as a potential pathogen. So even whether you're working with a coral bacterium or you're working with a human pathogen, you exercise the same cautions. We're all wearing gloves and lab coats, making sure you're not poking yourself with anything.
And just like you said, if you get cut, like out in the ocean, if you get cut by coral on accident, you don't ever want to touch coral because they are an animal. So we had some questions about this. So Blake, I know you touch the coral to work with it, and you have special permits and things to do so. But this isn't something that like anyone could just go out into the ocean and collect these coral specimens and bring them to their house and do experiments. So a lot of corals are some species of corals are quite endangered. So you definitely need to be careful. And as I said, coral is an animal, so you don't want to be touching it. You only brush up against it on accident, but if it does cut you, it does have this bacteria on its surface that could potentially give you a little infection. But it wouldn't affect us the same way it would affect corals, right?
I guess it depends on the bacterium. I don't think they've tested some of these on people. I don't think it's allowed. But it's one of the things like, it's better safe than sorry. So you treat everything. It's kind of the general rule here, is you kind of treat everything as a potential pathogen. So you never really want to find out.
Just be careful.
There's also a lot of other factors too. Some people might be more susceptible to infection than others. There's a lot of instances where you have seemingly harmless bacteria that can cause an infection in some unlucky people.
We have so many good questions. I know we're running onto the hour, so I want to just say, well, if you guys want to stick around, we'll take a few more questions. But if you have to run, I want to just say thank you so much for joining us for our last ever career dives. Well, not maybe ever, but for this year at least. So we'll take some more questions, but Blake, I want to ask you really quickly because we didn't get to address it. Did you want to share maybe one last nugget of wisdom for aspiring marine scientists?
Sure. My little nugget of wisdom is to stay curious. There's a lot, if you go into grad school, you tend to get caught up with, working on specific projects, and you may see something unusual and it's like, oh, well, I don't have time, or it's not related to my project, I'm not going to ignore it. But some of the coolest projects that I've done is because I've sat there and like said to myself, oh, that's weird. I want to know what's going on there. So, stay curious. I mean, that's the whole, it's kind of what we do as scientists. We're just curious people trying to figure out how things work.
Keep asking questions.
Mm-hmm.
Well, I want to say, Blake, do you have some time to stick around and answer a few more questions?
Sure, I'll be happy to.
Awesome. Well, I do want to mention, in case anyone has to go, thank you so much, Blake, for joining us and for talking about your career and your journey. If you enjoyed joining us and hearing about Blake's story and about the work he does, please stay tuned for our January program series, which is going to be called Marine Science in the Morning. We're going to be posting a lot about that on Facebook and a lot of our social media channels. I know Aaron is going to be posting some things in the group chat for you about links. We had a lot of teachers on this program and a lot of students, so I just want to mention that we do offer virtual field trips. So if that's something that you're interested in, please reach out to us at your convenience, but it's smseducation@si.edu and inquire more about those. We have virtual field trips for kindergarten through third through fifth, and then 6th, 7th, and 8th. Sorry, I'm struggling to remember. We have 3 different virtual field trips that you can sign up for on fish, mangroves, and we just did one on the carbon cycle. So if that is interesting to you, make sure to send us an email. And the links for all those things are going to be in the chat for you. What would also be really helpful, if you are a teacher and you had quite a few students, if you could email us and let us know how many, that would be amazing, but obviously at your convenience. So Blake, let's take some more questions because we have a ton coming in. So we answered questions about, the different types of diseases in coral and that, potentially they could make humans sick, but that we're not really sure. And I want to get to this question because I think it's really good. So Dr. Melidio's class, they just finished a whole unit on the oceans, which is really awesome. I hope you guys enjoyed learning more about oceans. And they learned about ocean ecosystems and interdependence. So could you tell us a little bit more about the impact on the ecosystem when corals get sick and get these diseases?
Oh, sure. So I guess one thing I need to point out is, the existence of disease in any system is natural. So to have a low level of disease or a certain level of disease is natural. What really messes up systems is when we have, unnatural outbreaks of disease that are, way beyond what, you normally see in a system. And, I use this metaphor a lot or analogy, comparison. I don't study English, apparently. This comparison, a lot of, you think of coral reefs as, like I mentioned before, is, the forests of the ocean, is that you have these physical structures that it's not just like what they're doing, but the actual physical structure of these coral reefs providing a shelter for a lot of organisms. And just like, trees, when the organ of the tree or the coral dies, that physical structure will start to degrade just from the natural processes of, decomposition in the ocean. Even though, coral skeletons are made out of like limestone, it will start to degrade over time. And you have the loss of these physical structures, and you just think of it as, a small little fish's home just kind of crumbling down. So, that has a tremendous effect on the ecosystem. You don't have a home anymore. You don't have a place for these little fish or organisms to hide from predators. You don't have areas for them to, have the little nurseries for the young juveniles to grow up in a safe place. There's a lot of, detrimental effects.
So if you think of it, like you said, just like if a lot of times the coral reefs get compared to rainforest, but if you lose the coral, it's synonymous with, like, imagine them just cutting down all the trees in the rainforest. All the animals that depend on that ecosystem and that habitat no longer have a place to hide or place to find food. So coral is super, super important to the marine ecosystems.
Yeah, and also something that people don't really think about is Corals actually benefit humans as well. They're involved with a lot of preventing erosion of shorelines, especially of like islands like Hawaii. They actually help prevent erosion, and also kind of lowers, stops long storm surges or strong storm surges, dissipates energy from those. So if you have a really nice healthy reef, it could potentially help protect you from things like hurricanes and coastal flooding and erosion. And and for people that live near the shore, that's really important.
Really, really important. And I think, I don't know if we maybe touched on this at all, but as you said, these bacteria and these disease outbreaks are natural, but there are certain things that are like making them happen at faster rates or spreading them a little bit faster than would normally happen, right?
Yeah, there's a few, there's multiple studies out there that show links between warmer ocean temperatures and disease outbreaks or sewage pollution. But also really, I mean, a combination of the biggest threat to coral reefs nowadays is climate change. There are disease outbreaks like the one in the Caribbean, stony coated tissue loss disease that's damaging a lot of different species, but also climate change as well. I mean, you have all your corals when they're weakened or stressed out. If you have a lot of higher water temperatures, that's too high, or you have pollution, sewage input, and things like that. So yeah, I mean, it's a combination of things as well. So you don't want to, I mean, and then you think about it, if something is less stressed, it might be able to fight off an infection better. There are a lot of great labs studying the immunology of corals because we don't really know much about its immune system. But it's kind of, I use this a lot too. It's like, well, if you had the flu and someone came up behind you and just slapped you, it's not really helping. It's kind of what's happening with the environmental conditions aren't very good if the coral that has an active infection, it's not going to help very much.
Yeah, definitely not. Blake, you got a lot of questions about what is the coolest coral? Have you ever seen a rare coral? What's the biggest coral?
What is the coolest coral? Oh, that's a little biased because I really like the rice coral that's in Hawaii. I mean, I worked with it for how many years? But I can tell you why it's cool. It has a very cool how it grows. So most coral, you have the skeleton, and on top of that skeleton is its tissue. So the rice coral, the tissue is throughout the skeleton. The skeleton is porous. So it's a very light skeleton. It's very brittle, but the tissue will grow into the skeleton. I guess the best example, I think of bone marrow. So if you look at the coral, you can see it's a very porous skeleton that grows into the tissue. The rarest coral, rare coral. There were a few colonies of an alien coral that ended up in Hawaii. I mean, I don't know if that counts as rare.
Supposed to be there.
Yeah. I've seen collections of black coral, which is a deep sea coral. I think hundreds and hundreds of meters deep. It wasn't alive, but it was just a sample. I guess that's
black. Is the skeleton black?
Hmm?
Oh, neat. Because usually they're white.
It's not. I don't think it's calcium carbonate like most of the hard corals. I forgot what it was made of. So I don't know. I don't study deep sea. Apologize to all the deep sea biologists. And then the largest coral. I've seen a lot of large corals.
Some of those ones in Palmyra Atoll were, you said larger than like 8 feet.
Yeah, even in Hawaii, some of the rice coral colonies were quite large. There was one that my coworkers referred to as Blake's colony because I used to do a lot of studies with it. I think that one is about 6 or 7 feet across.
Wow.
So this is quite a large colony. I've seen a lot of large colonies.
So it's rice coral, right? Like the food, rice.
Mhm.
Someone
Yeah.
asked earlier if you had a coral named after you, but it sounds like you have a colony of corals named after you.
Yeah, I haven't discovered a new species of coral. A few new species of bacteria. I don't know if I'd want to name a bacterium after me though. I don't know how I feel about that.
You can name it after me.
Oh, yeah.
Well, Blake, I want to say we're getting overtime, and I know that you are a very busy guy. So I want to wrap things up. I want to say thank you so much to everyone who joined us today, and thank you so much for all the incredible questions. We had a lot, and we might not have gotten to all of them, but if you do want to follow up with us at our email address, it's in the chat, but if you're on Facebook, it's smseducation@si.edu. And just to reiterate, like, this will live on our Facebook channel and will be on our YouTube channel in case you want to watch things back after the fact, if you had to pop away or if you couldn't hear a segment of it. I just want to make sure that everyone has access to it. Blake, thank you so much for joining us and for being our last career dives of 2020.
No problem. This is a great time.
Good. I'm glad you had fun. I did too. And I hope everyone listening did. Well, my friends, that's going to be it for me. I'm going to sign off. We'll look forward to hearing from all of our amazing panelists for our Marine Science in the Morning series starting January of next year. And we'll see you there.
All right. Bye.
Bye, everyone.